So good day class. So for today, we shall study the activities of the cell membrane or plasma membrane. Now before we go into the details of our lesson, let me just show you this electron micrograph of the cell membrane. So you will notice that the cell membrane is made up of two layers. So we will go into the details of that later. Now the cell membrane or the plasma membrane is flexible and elastic, and the cell membrane allows protozoans like the one in this picture to move. So the cell membrane plays a very important role in the locomotion of certain organisms like protozoans. Also, the cell membrane plays an important role in homeostatic balance. So the cell membrane maintains balance or equilibrium by controlling what goes in and what goes out of the cell. The plasma membrane maintains balance by controlling what enters and leaves the cell. So let us now go into the details of the different functions of the cell membrane or plasma membrane. Now, the cell membrane serves as a protective barrier. Now, what does this mean? Now, just like the outer layer of your skin or the epidermis that separates your body from the environment, the cell membrane separates the inner content of the cell from its exterior environment. This cell membrane provides a protective barrier around the cell and regulates which materials can pass in and out. So that's what we meant by being a protective barrier. And just like what I mentioned a while ago, the plasma membrane plays a very important role in homeostatic balance. The cell membrane or the plasma membrane regulates the entry and exit of substances. And this property of the plasma membrane regulating what goes in and what goes out of the cell is known as selectively permeability or semi-permeability. Alright, so the cell membrane also allows cell-to-cell recognition. Now what does that mean? Now the cell membrane being a barrier helps protect the contents of the cell from the outside environment, and also cell recognition, which is a way for the body to communicate. It is a critical part of the immune system where several mechanisms help recognize other cells, self-cells as well as foreign materials such as bacteria, viruses, and toxins. So it understands which to protect and which to destroy. So that's what we meant by allowing cell recognition. Now, aside from allowing cell recognition, the plasma membrane also performs the following. The cell membrane provides anchoring sites for filaments of the cytoskeleton. The plasma membrane also provides binding sites for enzymes. The cell membrane also provides interlocking surfaces that bind cells together like in gap junctions. And the cell membrane contains the cytoplasm, which is the fluid in the cell. Alright, so here is an illustration showing the structure of the cell membrane or the plasma membrane. So you will notice that the cell membrane is made up mostly of the following molecules. So we have phospholipids. Also there are cholesterol molecules embedded in the cell membrane. Then also we have proteins, either integral proteins and peripheral proteins. And also we have carbohydrates, which plays an important role in cell recognition. So again, here is an illustration of the structure of the cell membrane. So again, you will notice that the cell membrane is made up mostly of this molecule called phospholipids. A phospholipid molecule contains a head, and also it contains two fatty acid chains that are non-polar, and the head, like what I mentioned, is polar and contains a phosphate group attached to a glycerol component. So here is a diagram showing the structure of a phospholipid molecule. Now the phospholipid molecule has a head. Now the head contains a phosphate group which is attached to a glycerol component. The head of the phospholipid molecule is polar, so meaning it's hydrophilic and it attracts water molecules. Now attached to the head are two fatty acid tails. The two fatty acid tails are non-polar, so they repel water, or the two fatty acid tails are said to be hydrophobic. Alright, and the cell membrane follows this model known as the fluid mosaic model. Now what does this mean? Now the word fluid means that the individual phospholipids and proteins can move side to side within the layer like it's a liquid. And when we speak of the word mosaic in the fluid mosaic model, the pattern produced by the scattered protein molecules when the membrane is viewed from above, so it would look like a mosaic. Alright, so here is an animation showing the side-to-side movement of these protein molecules that are embedded in the phospholipid bilayer. So they move side to side like it is in a liquid. And if you look above, the scattered proteins would resemble a mosaic. Hence the term fluid mosaic model. Alright, so going back to the phospholipid molecules. So the phospholipid molecule is made up of a head which is polar. And the polar heads are said to be hydrophilic or water-loving. And the two fatty acid tails are non-polar and they are said to be hydrophobic or water-fearing. And this property of the phospholipid bilayer, if you will notice, there are two layers of phospholipid molecules. This makes the membrane semi-permeable, or it makes the membrane selective in what process. So this allows the regulation of the entry and exit of substances. So that's the phospholipid bilayer of the cell membrane. So there are two layers of phospholipids. The head of the phospholipid molecule is polar, so it is hydrophilic, it attracts water. And the two fatty acid tails are non-polar, or they are hydrophobic, or they repel water. So like what I have mentioned, the cell membrane is made up of two layers of phospholipid molecules, as you could see in this illustration. The cell membrane is made up of two layers of phospholipids, hence it is called a lipid bilayer. Now the question is, what are the molecules that can pass through the phospholipid bilayer and what are the molecules that cannot? Now, hydrophobic molecules can pass easily, while hydrophilic molecules do not. Materials that are soluble in lipids can pass through the cell membrane to the phospholipid layer quite easily. Now, molecules, small molecules, and larger hydrophobic molecules move through easily through the phospholipid bilayer like oxygen and carbon dioxide. Now, in the case of water, now we know that water is a polar molecule. And technically, water cannot pass through the phospholipid bilayer easily. Now, in the case of water, there are special channels embedded in the phospholipid bilayer called aquaporins. These aquaporins, these are open channels that will allow the entry of water. So, water can pass through easily through those channels called aquaporins. Now, molecules such as ions, hydrophilic molecules larger than water, and large molecules like proteins cannot pass through the phospholipid bilayer on their own. So in order for these molecules to enter the cell, they need to enter through special channels embedded in the phospholipid bilayer. So we will be discussing those special channels later. Now at this point, let us now study the types of transport across the plasma membrane. Now molecules move through the cell membrane by the following mechanisms. So we have passive transport of molecules across the cell membrane and active transport. Also we have what we call bulk transport of molecules. Now under passive transport, we have diffusion. Now under diffusion, we have osmosis, facilitated diffusion, and simple diffusion. Alright, so let's talk about now diffusion. Now basically when we speak of diffusion, diffusion is a type of passive transport. That means that no energy is required to move the molecules across the membrane. So the molecules, they move through kinetic energy, natural kinetic energy. So these molecules move in the direction from higher concentration to lower concentration. In simple diffusion, there is no energy required, and molecules move through natural kinetic energy, meaning that molecules move from a region of high concentration to low concentration. So meaning that these molecules, they move down the concentration gradient. Example of this would be oxygen or water diffusing into a cell and carbon dioxide diffusing out. Now here is an example to illustrate simple diffusion of molecules. So here again, in simple diffusion, there is no energy required. There's no need for ATP or adenosine triphosphate to move these molecules from a region of high concentration to a region of low concentration. There's no energy required. Molecules simply move down the concentration gradient. Take note of the term that I'm using. The molecules moves down the concentration gradient. When we say concentration gradient, there is a difference in concentration. And these molecules move from a region of high concentration to low concentration. So based on this animation, so you will notice here that the molecules, they tend to spread out evenly in solution. They move from a region of high concentration to low concentration, and the movement of these molecules will stop until equilibrium is achieved. So that is one way of maintaining balance. So this movement of molecules from high to low concentration will continue until equilibrium is achieved. So here is another example to further illustrate simple diffusion. This time this is diffusion of liquids. So here we have dye molecules that are applied to this beaker filled with water. Now the dye molecules will tend to spread out evenly in solution from a region of high concentration to a region of low concentration. So in other words, the dye molecules will tend to spread out evenly in solution as seen here in this photo labeled B. So the molecules will tend to spread out evenly in solution, moving from a region of high concentration to low concentration, or the molecules will move down the concentration gradient. And the movement of the molecules will only stop until all the molecules are spread out evenly in solution, or in short, equilibrium or balance was achieved. So class, I hope everything is clear about simple diffusion. Here is another example to illustrate simple diffusion. So let's say this is a bowl of soup. So the molecules of soup will tend to spread out evenly across the room from a region of high concentration to low concentration. So the molecules will tend to spread out evenly, and eventually in a few minutes you would smell the delicious scent of that soup across the room. So the molecules will only stop diffusing until balance or equilibrium is achieved. Alright, so that is diffusion, simple diffusion. Again, molecules here, they don't require energy to move from a region of high to low concentration because the molecules are moving down the concentration gradient. Molecules move from a region of high concentration to a region of low concentration. So molecules, they move down the concentration gradient. Now what about osmosis? Now osmosis is a special type of diffusion. Now this is diffusion of water. Take note, diffusion of water across a semi-permeable membrane. And molecules move just like in simple diffusion from a region of high concentration to a region of low concentration. But here we only focus on the diffusion of water across a semi-permeable membrane. Now class, let's take a look at this example to illustrate the mechanism of osmosis. Now we have here a U-shaped test tube. Now at the middle of this U-shaped test tube is a semi-permeable membrane. The semi-permeable membrane will allow only water molecules to enter, and this membrane will not allow solutes to penetrate, like in the case here we have, let's say, sugar as our solute. Now let us divide the U-shaped test tube into two. We have side A here on the left side and side B here on the right side of the U-shaped test tube. Now here in side A, we have here a higher concentration of solvent. That's water. Our solvent is water and our solute is sugar. So here we have less solute, less sugar, and more of the solvent, more of the water. Alright? In side B, you have less water, less solvent, and more of the solute. There are more sugar molecules. So this is a U-shaped test tube that is not in equilibrium or balance. So here we don't talk about the movement of the solute molecules, since the solute molecules cannot penetrate the semi-permeable membrane. But here water can. And here we can see a difference in the concentration of water. There's a higher concentration of water here inside A and less concentration of water inside B. So there is now a concentration gradient or a difference in concentration. So naturally, water molecules will move from a region of higher concentration to lower concentration. So water will move from side A to side B. So you will notice here that the water level inside B begins to rise. And the movement of water molecules will only stop until balance or equilibrium is achieved. Alright, so that's how we explain osmosis through this U-shaped test tube. Again, in osmosis, we focus only on the movement of water molecules through a semi-permeable membrane. So again, in this animated illustration, you have here side A and side B. Alright, this setup is separated at the middle by a semi-permeable membrane. Let's say side A here is on the left side and side B on the right side. On side A here, we have a low concentration of sugar molecules and a higher concentration of solvent or water. Here in side B, you have a high concentration of solute or sugar and a low concentration of solvent or water. So here we have a concentration gradient. There is a higher concentration of water in A and a low concentration of water in B. So water moves from a region of high concentration to low concentration, or water moves here from side A to side B. So you will notice that the water level here in side B rises, and this rising of water level will stop only until balance of the system is achieved. So that is osmosis, class. Osmosis involves the movement of water across a semi-permeable membrane. Now, I think we mentioned something about aquaporins a while ago. Alright? So again, water moves from a region of high concentration here to low concentration. And water moves through special channels called aquaporins because water is a polar molecule, and technically water cannot pass through the lipid bilayer of the plasma membrane. So there are special channels called water channels or aquaporins. These aquaporins, they allow water to enter the cell. These are protein pores, and they are used during osmosis of water. So water enters the cell through these special channels called aquaporins. So at this point, class, let us now apply the concepts of diffusion and osmosis in the maintenance of balance in both the external environment of the cell and the internal environment of the cell. Now let's take a look at this illustration. So we have here the blue area, that is the external environment of the cell, and we have the yellow area, that's the internal environment of the cell or the cytoplasm. Now, the external and the internal environment of the cell is separated by the plasma membrane or the cell membrane. Now, everybody kindly take a look at the concentration of solute and solvent outside the cell or the external environment. So, we have here 10% solute, in this case salt NaCl. So, you have here 10% NaCl and 90% solvent or water in the external environment. Now let's go to the internal environment of the cell. So you have 10% NaCl and 90% water as well. So you will notice, class, that both solute and solvent in and out of the cell are equal. So that means that the cell is in homeostatic balance with the external environment. So that means there is no net movement of water. So the cell is said to be in an isotonic solution. The cell is said to be in homeostatic balance. Now let's consider the next case. Now the cell here is immersed in a hypotonic solution. Now what do we mean by a hypotonic solution? Now hypo means less. So if you will take a look at the percentage of solute and solvent outside the cell, there is here 10% salt and 90% water. Compare that to the amount of solute and solvent inside the cell. So inside the cell, you have 20% salt or solute and 80% of the solvent or water. So here you will notice that there is less solute or salt outside the cell. So that's what we call a hypotonic solution. Hypo means less of the solute outside the cell compared to the amount of solute inside the cell. So you have here 10% salt outside and 90% water outside compared to the water inside the cell, which is 80%. So there's more water outside the cell. So we have here a system wherein there's no balance or equilibrium. So in order to achieve balance or steady state, water moves from a region of higher concentration to a region of lesser concentration. So water tends to get in the cell. So what is the direction of water movement? Water moves in the cell. And as the water continues to move in, the cell begins to swell. And eventually, if the process continues, the cell will cytolyze. Cytolysis means cell bursting. Alright, so that's what happens if the cell, especially for animal cells, if the animal cell is immersed in a hypotonic solution. But this is not the case for the plant cell because the plant cell is surrounded by a cell wall. The plant cell will just swell up, pressure builds up inside the plant cell, and that's what we call Turgor pressure. So the plant cell, when immersed in a hypotonic solution, the plant cell becomes turgid. But for animal cells, the cell will swell and eventually the cell will cytolyze. Now, what about this next case? So in this next case, we have here the cell immersed in a hypertonic solution. So kindly take a look at the amount of solute and solvent inside and outside the cell. So outside the cell, we have 15% salt compared to 5% salt inside the cell. And 85% water outside the cell compared to the water inside the cell, which is 95%. So hyper here, you have more of the solute outside the cell compared that to the amount of solute inside. So more solute outside, that makes it hypertonic. So more solute outside, less solvent outside, and more of the water inside the cell. So there is no balance. Now water here in a hypertonic solution tends to move out, no, from a region of higher concentration, 95% water inside, to a region of lesser concentration, 85% of the water outside. So water tends to move out. What is the direction of water movement? Water moves out of the cell. So water moves out, the cell loses water, eventually the cell shrinks. Alright, and eventually the cell will plasmolyze, or what we call cellular shrinking, plasmolysis. But again, for the plant cell, since the plant cell is surrounded by a cell wall, the vacuole simply collapses. Alright, and the cell or the plant becomes flaccid. No? Turgor pressure loses, so the cell becomes flaccid. Alright class, so here are blood cells immersed in a hypertonic solution. So we can see here the plasmolysis or the crenation of the cell. Alright, so here are plant cells immersed in a hypertonic solution. The vacuole collapses, the cytoplasm collapses, but the cell wall remains intact. Alright, so again, as a review, in a hypotonic solution, water moves in. As you could see in our diagram, water moves into the cell. So the animal cell swells and eventually it cytolyzes. If the cell is immersed in a hypertonic solution, water moves out of the cell, then the cell shrinks or the cell plasmolyzes. And if there is equal concentration of solute and solvent in and out of the cell, there is no net movement, so the cell is in an isotonic state or homeostatic state. So class, let's take another look at these three extracellular environments. Now, if the cell is immersed in an isotonic solution, there's equal amounts of solute and solvent in and out of the cell, then the result would be that there is no net movement of water molecules. And that means that the cell is in a state of balance or steady state. Now, if the cell is immersed in a hypotonic solution, that means that there's more water outside the cell. The water moves in the cell and eventually the cell swells and the cell will burst or it will cytolyze. The result is cytolysis. Now, if the cell is immersed in a hypertonic solution, water is less outside the cell. Water is greater in concentration inside the cell. So water moves out from a region of greater concentration to a region of lesser concentration. So water moves out of the cell. So the cell shrinks. And that's what we call plasmolysis. So for the plant cell, again, because of the cell wall, only the vacuole and the cytoplasm shrinks but the cell wall remains intact. So there is loss of pressure, so the plant becomes flaccid in a hypertonic solution. In a hypotonic solution, if the plant cell is immersed in a hypotonic solution, and again because of the cell wall, pressure builds up inside. As water enters, pressure builds up. So that's what we call Turgor pressure. The plant becomes turgid. Alright, so those are the three extracellular environments and we connect that with the principles of osmosis and diffusion. Alright, so before we move on, let's watch this short video about diffusion, osmosis, and the three extracellular environments: isotonic, hypertonic, and hypotonic solutions. Alright class, let's watch this.
Cells have a very finite range of conditions in which they can survive. If it's too hot, too cold, too watery, too salty, too acidic, or too basic, they can't function properly. We're going to explore what happens when you place a cell in a solution that is much more salty, hypertonic, much more watery, hypotonic, or has an equal salt concentration, isotonic. Remember that cell membranes are selectively permeable. They allow certain particles to pass through, but not others. Usually, larger molecules can't fit through the membrane without special channels. The cell membrane is fitted with special protein channels called aquaporins, which allow water molecules to pass through without expending energy. However, the ease with which the water molecules can cross is going to depend on something called the concentration. Concentration is a measure of how much solute there is per volume of solvent. In other words, whether the liquid is more salty or more watery. We'd usually expect that the molecules would follow a process called diffusion. They would flow from areas of higher to lower concentration. Eventually, they will reach equilibrium, where there will be an equal concentration on both sides of the membrane. However, in the case of a liquid like salt water, the salt molecules are much too big to fit through the membrane, so the only particle that can move is water. Let's look a little more closely. Water molecules are highly attracted to salt molecules. They cluster around salt and really don't want to move. The positive parts of the water molecule stick to the negative parts of the sodium chloride, and vice versa. The interactions between oxygen and sodium and between hydrogen and chlorine are called ion-dipole interactions, which you might remember from chemistry. Water molecules that are not stuck to a molecule of salt are far more likely to relocate. Today we're going to immerse a cell in 3 kinds of liquids: a very solute-rich liquid, a very watery liquid, and a liquid that's in between. Then we'll see what happens to the water molecules. In this first scenario, the liquid surrounding the cell has many molecules of solute in it. This liquid is hypertonic compared to the cell. It has a much higher concentration of solute particles and a much lower concentration of water. Unfortunately, the solute molecules can't pass through the membrane to reach equilibrium, but water molecules can. The molecules of water on the outside of the cell are going to be obstructed from passing through by the many molecules of solvent. However, the molecules on the inside have much less solvent getting in the way. Water will begin rushing out of the cell. Some water will come in, but the net movement, the overall movement, of the water molecules will be outwards and the cell will shrink. We call this cell contraction plasmolysis. This is also how pickles are made. Now let's immerse a cell in a much less concentrated solution, a hypotonic solution. Compared to the cell, it's much less salty. Some water molecules will exit the cell, but even more will rush in because they aren't obstructed by solute particles. The cell grows in size and it may even undergo cytolysis and burst. Placing a cell in an isotonic solution, where the concentrations inside and outside are equal, is much more pleasant. Equal amounts of water molecules continue to pass in and out of the cell. The system is in dynamic equilibrium. Particles are continuing to move, but the net movement is zero. The cell neither grows nor shrinks and is much more likely to survive. Now let's return to our seawater question. Seawater is incredibly salty, much more salty than our body's cells. Since it's such a hypertonic solution, if you immersed your cells in it, they would shrivel up and die. The real danger, however, lies in how your kidneys would react to the seawater. Your kidneys would attempt to remove the toxic levels of salt by using water pulled from your cells. Your body would actually use more water in removing the salt than was originally contained in the seawater itself. In summary, drinking seawater is a lousy idea even in extreme circumstances. You're better off drinking your own pee. So there we have it, hypertonic, hypotonic, and isotonic solutions, as well as a couple of practical examples.
Okay, so the topic of this video is to kind of compare and contrast solutions that are hypotonic versus ones that are hypertonic. So let's get started. Okay, well, first of all, what is a solution? Well, it's a mixture where one substance will dissolve in another. And a solution generally has 2 parts to it. There's the solute. It's the parts that are actually dissolved. And then here's some solutes in my picture here. The S's are symbolic of the solutes. And then there's the solvent. Now, in a biological solution, that solvent is typically water. So here I have in my animation here, I have solutes and water mixed together. Well, let me ask you, what makes sense? Does it make sense to say that sugar dissolved in the water or water dissolved in the sugar? I hope it makes sense to say that the sugar dissolved in the water. Because the sugar dissolved, it's the solute. The water caused the sugar to dissolve, so the water is the solvent. Well, here's a cell. And if we zoom on into this area right here in the cell membrane, we see the phospholipid bilayer, the vertical cell membrane right there. And this is our cell membrane, or our plasma membrane. You might know that it is semi-permeable, meaning that certain molecules can pass freely, but others cannot. Well, let me add some water molecules on both sides of the plasma membrane, the cell membrane. And let me add some solute molecules to both sides of the plasma membrane. Now, when we say the plasma membrane is semi-permeable, so water is able to pass through the cell membrane. Now, if you remember osmosis, the movement of water from high to low concentration. So water will generally move from the higher concentrated area to the lower concentrated area, but water is able to pass from one side of the cell membrane to the other. But molecules like the solutes, for a variety of reasons, are incapable of passing through our cell membrane here. And for a variety of reasons, such as sometimes solutes are too large, sometimes solutes have a charge on them that make them unable to pass through the cell membrane here. So the plasma membrane generally can allow water to pass freely, but not the solutes. Okay, so let's go ahead and talk about a hypotonic solution now. It's one with a lower concentration of solutes, and that is contrasted to a hypertonic solution, one with a greater concentration of solutes. In my picture here, I've got a beaker filled with a fluid and a over-exaggerated size of a cell on the inside. Let's pretend the solution in the beaker is 80% water, 20% solute. But the solution in the cell is 60% water, 40% solute. Well, where is the hypotonic solution? You have two choices. Which solution is hypotonic? The definition of hypotonic is one with a lower concentration of solutes. Therefore, the solution in the beaker in this example is hypotonic because the 20% solute is a lower concentration than the 40% solute in the cell. And which solution is hypertonic? By definition, hypertonic is one with a greater concentration of solutes. That would be the solution in the cell, because the 40% solute in the cell is greater than the 20% solute in the water outside the cell. So now that I've labeled the solutions outside of the cell as hypotonic and the solution inside of the cell as hypertonic, I want to ask you, what would happen to a cell that is placed in a hypotonic solution? This cell right here is placed in a hypotonic solution. Well, we remind ourselves the definition of osmosis. It's the movement or the diffusion of water from a high concentration to a low concentration. Well, where is the high water concentration at? If you look at the picture, you can see 80% is a higher percentage than 60%. So water would diffuse or enter into this cell over time. Over time, the cell would begin to swell and get larger and larger and larger to the point where it actually could even burst depending upon how much water flowed into it. Well, let's look at this situation right here. Let's change the concentration a little bit. Now pretend the solution in the beaker is 50% water, 50% solute. But the solution in the cell is still the same, 60% water, 40% solute. Well, what solution, which of these, choice A or choice B, which solution would be considered hypotonic? Hypotonic is one with a lower concentration of solutes. So therefore, the solution in the cell is now considered the hypotonic solution because the 40% is a lower percentage than the 50%. And which solution would be considered hypertonic? By definition, hypertonic is one with a greater concentration of solutes, and that's the solution in the beaker. 50% is a greater percentage than 40%. So now that I've labeled this solution as hypertonic, the solution in the beaker is hypertonic in this example. what would happen to a cell placed in a hypertonic solution? Again, we remind ourselves the definition of osmosis, the movement to the diffusion of water from a high to low concentration. Where is the higher water concentration at? Look in the picture. I see that 60% is a higher percentage than 50%. So therefore, water would actually diffuse out of this cell. It could start to shrivel and lose kind of some of its water mass as a result. Okay, well, here is our cell again, and like we saw before, let's go ahead and zoom into this area of the cell membrane. And when we do zoom into this area, we can go ahead and again label the cell membrane, their plasma membrane, as being semi-permeable. And let me add 15 water molecules inside the cell, and let me add 5 solute molecules inside the cell. On the outside, let me go ahead and add 5 water molecules, and let me add 5 solute molecules. Okay, so now that I set up this situation, where do these two labels go? Does the hypotonic label go on the left or right? Does the hypertonic label go on the left or right? Well, look at the percentages. 15 waters, 5 solutes, that's a total of 20 molecules. So we have 75% water solution, 25% solute solution. On the right-hand side, we have a 50-50 solution, 50% water, 50% solute. So now I can go ahead and label the solution on the left as hypotonic and the one on the right is hypertonic. Hypotonic on the left. Hypotonic is a solution with a lower concentration of solutes. 25% is a lower concentration than 50%. Now, what would happen over time? Remember, what does water do over time? Through osmosis, it will diffuse. Watch the water on the left. It will diffuse slowly over time until there's an equilibrium, a balance. Well, let's re-add our percentages. Now there's only 10 water molecules on the left. That would be 67% water, 33% solute. Now what about the right-hand side? Over time, now that diffusion and osmosis has occurred, there are now 10 water molecules on the right-hand side. That would also be a 67% water, 33% solute. Notice how they're equal to one another. Neither of these are hypotonic or hypertonic anymore. Once an equilibrium and a balance has been established, this is what an isotonic solution is. And so if you've seen the word hypotonic, hypertonic, and isotonic in cooperation with one or combination with one another, I hope that explains what those are. So as we wrap up this video, if you're in my biology class, pause the video and try to answer these questions on a separate sheet of paper. And I hope you found this helpful. Thanks for watching.
Alright class, so I hope those 2 videos that you saw had helped you gain more understanding of the concepts of diffusion, osmosis, and the 3 types of extracellular environments: hypertonic, hypotonic, and isotonic solutions. So at this point, let us now proceed with the other type of passive transport, which is facilitated diffusion. Now, from the word itself, facilitated means to assist or to help. So diffusion means that the molecules still move down the concentration gradient from a region of high concentration to a region of low concentration. So this is still diffusion. So molecules still move down the concentration gradient. But facilitated means that there are channel and carrier proteins, transport proteins that facilitate or assist the movement of these molecules down the gradient. Now, there are 2 types of these transport proteins that are embedded in the cell membrane to facilitate the movement of these molecules through facilitated diffusion. So we have channel proteins that are embedded in the cell membrane, which have a pore for materials to cross. And we have carrier proteins that can change shape to move materials from one side of the membrane to the other. So these transport proteins, they facilitate the movement of molecules down the gradient from a region of high concentration to a region of low concentration. So here is an animated example of such transport protein. So this is a channel protein. So channel proteins, they have a pore that allows the movement of molecules across the cell membrane. Molecules will randomly move through pores in channel proteins. Like for example, aquaporin. Aquaporin is an example of a channel protein. Alright? And we have carrier proteins. Carrier proteins, they change shape that allows molecules to cross the cell membrane. So that is facilitated diffusion. So these carrier proteins, they change shape so that molecules can move down the concentration gradient. Alright? So some carrier proteins do not extend through the membrane. They bond and drag molecules to the lipid bilayer and release them on the opposite side. Carrier proteins, they change shape to move materials across the cell membrane. Alright, now another type of transport mechanism across the membrane is what we call active transport. In active transport, energy is required in the form of ATP. So energy is needed in the form of ATP in order to transport molecules across the membrane from a region of low concentration to a region of high concentration. That means that the molecules will move against the concentration gradient. This is called an uphill movement of molecules from a region of low to high concentration. So let's take a look at this animated illustration showing active transport. So just like in facilitated diffusion, active transport involves transport proteins. But unlike facilitated diffusion, active transport uses energy from ATP. These transport proteins uses or requires energy from ATP in order to transport molecules from a region of low concentration to a region of high concentration. In short, these molecules are moving against the concentration gradient. It is like the molecules are being pumped from a region of low concentration to high concentration. The molecules are moving uphill. This is an uphill transport of molecules, or the molecules are moving against the concentration gradient. And one good example of an active transport of molecules across the membrane is the sodium-potassium pump. The sodium-potassium pump pumps sodium ions out and potassium ions in against the strong concentration gradient. So let's take a look further at the sodium-potassium pump. So let's take a look at this animated illustration. So in the sodium-potassium pump, you have here transport protein similar to facilitated diffusion. But in this case, the transport protein requires energy. And that energy from ATP changes the shape of the transport protein so that it pumps 3 sodium ions out of the cell and 2 potassium ions in. Right? So as you could see in this animation, 3 sodium ions are being pumped outside the cell and 2 potassium ions in. So 3 sodium ions out, right? Energy is required to change the shape of the transport protein so that 3 sodium ions exits and 2 potassium ions enters the cell. 3 sodium out and 2 potassium ions in. Alright, so eventually you have here an accumulation of sodium ions outside the cell, and there is an accumulation of potassium ions inside the cell. So this is an example of active transport, the sodium-potassium pump. So 3 sodium ions pumped in for every 2 potassium ions pumped out of the cell. And in the case of the nerve cell, this creates a membrane potential. But anyway, we will go into the details of action potential later. So before we continue with bulk transport of molecules, let's watch this short video about the sodium-potassium pump. Alright class, let's watch this.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
Alright class, so I hope everything is clear about the transport of molecules across the membrane. We have passive transport. Under passive transport, we have diffusion, osmosis, and facilitated diffusion that doesn't require energy, and molecules move down the concentration gradient from a region of high concentration to a region of low concentration. Then we have active transport of molecules that requires energy in moving molecules against the concentration gradient from a region of low to high concentration. So at this point, let us now study what we call bulk transport of molecules. There are 2 types of bulk transport of molecules. We have exocytosis and endocytosis. Moving the big stuff, that's bulk transport. So in exocytosis, molecules are moved out of the cell by vesicles that fuse with the cell membrane. This is how many hormones are secreted and how nerve cells communicate with one another. So that is exocytosis, moving the big stuff out of the cell. So again, you have here the vacuole that contains the substances that needs to move out. So the vacuole fuses with the membrane and the materials inside the vacuole moves out of the cell. Alright, so here is another illustration of exocytosis. So you have here the vacuole that contains the molecules that are needed to move out. The vesicle or the vacuole fuses with the cell membrane, and then the substances that needs to go out exits the cell. So that is exocytosis, moving the big stuff out of the cell. Again, here you have a vesicle containing the substances that are needed to exit the cell. So the vesicle fuses with the cell membrane, and the materials that needs to go out eventually exits the cell. Alright, So that is exocytosis. Then we have endocytosis, moving the big stuff inside the cell. So here you have the formation of a vesicle. So you have here the cell membrane forming an invagination surrounding the material that needs to get in. Then eventually the material is surrounded by a vesicle or a vacuole. Then the vacuole or the vesicle pinches off and moves in the cytoplasm. Alright, now another type of endocytosis, a special type of endocytosis, is known as Spinocytosis. This is the most common form of endocytosis wherein dissolved molecules are taken in. Alright, or what we call cellular drinking. So the cell forms an invagination like what I mentioned earlier. Materials dissolved in water is to be brought into the cell. So pinocytosis is also known as cellular drinking.
Cells transport material in and out across their cell membrane, which is a barrier made up of a double layer of lipids with embedded protein and carbohydrate components. Some molecules can diffuse across the membrane, or be transported across with the help of membrane-bound proteins. For transport of larger cargo, however, cells use endocytosis and exocytosis to transport material in and out of the cell, respectively. There are roughly 5 categories of molecule that try to get across the cell membrane. Small nonpolar molecules like oxygen or carbon dioxide are able to diffuse rapidly through the cell membrane. Small polar molecules like water can cross as well, but do so very slowly. Large nonpolar molecules like vitamin A are also very slow to cross the cell membrane. And large polar molecules like glucose and highly polar charged ions like sodium, potassium, and chloride, as well as molecules that possess a charge like amino acids, are all highly unlikely to get across a cell membrane on their own. So, many of these molecules, some common ones being water, glucose, and ions, pass through the membrane using transport proteins. Examples of transport proteins include channels like aquaporins, which is a water channel, and chloride channels, which let chloride ions cross membranes, as well as carriers such as the glucose transporter. However, when the cell needs to transport a lot of molecules or one very big molecule, it resorts to bulk transport, which comes in 2 styles: endocytosis and exocytosis. Endocytosis is a process that cells use to engulf extracellular material, and exocytosis is the opposite of that, during which cells expel material into the extracellular space. Both endocytosis and exocytosis need energy in the form of adenosine triphosphate, or ATP, used in the movement of the substances in and out of the cell. There are 3 types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis, where phago means to eat, is used by white blood cells like macrophages and neutrophils, which patrol the body looking for debris, bacteria, and dead cells to eat. So let's imagine that a macrophage comes across a particularly bothersome streptococcus. First, the strep attaches to macrophage receptors on its cell surface. The macrophage then extends arm-like projections called pseudopods around the strep, like a death hug. Then the strep is slowly engulfed by the cell membrane, which invaginates to form a vesicle on its inner side. The vesicle then separates from the cell membrane, forming a phagosome. During this step, an electron pump uses ATP to pump protons into the phagosome, lowering the pH inside. In the cytoplasm, the phagosome encounters an organelle called a lysosome, which contains digestive enzymes. The lysosome and the phagosome fuse together, merging their contents, forming a structure known as the phagolysosome. Inside the phagolysosome, lysosomal enzymes start destroying the bacteria with the help of an acidic pH. After it's all over, lysosome heads over to the cell membrane to expel the leftovers out into the extracellular space, like a cellular burp. Pinocytosis, on the other hand, literally means the cell drinks. In pinocytosis, the cell's plasma membrane invaginates to form a small cup around the portions of extracellular fluid and solutes that are not dissolved in it. Then, the edges of the cup come together, forming a vesicle. Since the cell is not really eating anything other than the occasional solute, the result is not a phagosome, but merely a vesicle. The pinocytosis vesicle is much smaller than a phagosome. Also, unlike phagocytosis, pinocytosis is a non-specific way for cells to take in solutes. So, whatever solutes are hanging around in the extracellular fluid get pulled inside the cup as well. And once inside the cell, motor proteins like kinesin or dynein carry the pinocytosis vesicle using ATP deeper into the cytosol. At the same time, the vesicle slowly releases the extracellular fluid and the solutes into the cytosol as well. Now finally, sometimes endocytosis involves special receptor proteins on the cell membrane, so it's called receptor-mediated endocytosis. Some molecules are taken in this way, like transferrin, which is an iron-binding protein, or low-density lipoproteins, or LDL, which contain cholesterol. As an example, let's see how this goes for LDL. Now, on the surface of the cell membrane, there are indented pits that have specific receptors for molecules like LDL. These pits are covered on the intracellular side of the cell membrane by a layer of clathrin proteins, so they're also called coated pits. Now, let's say LDL binds to its receptor in one of these pits. The edges of the pit start coming together. At the same time, the clathrin proteins inside the cell link up to one another like a sturdy shell around the forming vesicle. Once the vesicle pinches off from the cell membrane, the clathrin proteins detach from it and go back to the cell membrane. Inside the cell, the vesicle merges with an organelle called the endosome. Endosomes are similar to lysosomes in that they also fuse with ingested vesicles, but they can also do something else. Endosomes can separate the LDL particle from the LDL receptor it bound to. This is because the endosome has a proton pump that uses ATP to generate a low pH within, which causes the LDL to separate from the receptor. At that point, the vesicle splits into two vesicles, one which has all of the LDL that's been brought into the cell, and the other that has all of the LDL receptors. The LDL-filled vesicle goes to the lysosome for digestion, while the one containing the receptors goes back and releases the receptor back on the surface of the cell membrane. This is called receptor recycling because now the LDL receptor can bind to another LDL molecule and the cycle can repeat itself. It's kind of like washing and reusing a dish instead of buying new plates for every meal. Now, exocytosis, on the other hand, starts deep within the cell, in an organelle called the Golgi Apparatus. This organelle takes the proteins, lipids, and hormones that are generated in both the rough and smooth endoplasmic reticulum and packages them into a vesicle that can be zip-lined around the cell using the cytoskeleton. The cytoskeleton is made out of proteins like microfilaments, microtubules, and intermediate filaments, which all provide structural stability. The cytoskeleton is also very dynamic, allowing the cell to change shape by selectively extending and contracting the filaments, which is important in some cell functions like muscle contraction, cell division, and even cell movement. The cytoskeleton also helps structures within the cell move from one area to another. Now, there are secretory vesicles which move molecules out of the cell with help of motor proteins like kinesin or dynein, which pick up the vesicle and carry it towards the cell membrane along microtubules using ATP as fuel. The vesicle moves toward the cell surface, fuses with the cell membrane, and ruptures on its external side, spilling its contents into the extracellular space. Alright, as a quick recap. Endocytosis refers to the process in which cells engulf extracellular material, and there are different forms of endocytosis like phagocytosis, pinocytosis, and receptor-mediated endocytosis. Exocytosis refers to the process in which cells expel material into the extracellular space. Both endocytosis and exocytosis require ATP in order to happen.